Good afternoon. I'd like to start by thanking our host, the Lejeune Foundation, and thank you, Mark, for inviting me to this lovely city with this lovely weather and this lovely time of year. Today I'm going to talk about Alzheimer's disease in older adults with Down syndrome, which you have just heard quite a bit about. This will be a little bit more and slightly different in orientation. My focus is going to be on diagnosis of dementia, mostly. So, I come from Johns Hopkins University in Baltimore. This is where I work, in only one little building hidden behind all of this red brick. Disclosures, I have a couple. The big one being the fact that our research is funded by the Eunice Kennedy Shriver National Institute on Child Health and Human Development. Whoops. So the overview, I'm going to tell you a little bit about Alzheimer's disease, sort of the bird's eye view. I'm going to then review the connection between Alzheimer's disease and Down syndrome, some of which Dr. Lott just described. I'm going to give you a general description of our own program on aging and dementia in adults with Down syndrome, and then show you some selected findings focused on diagnosis, quantification of progressive decline, and then a little bit of biomarkers, and then hopefully time will allow me to chat a little bit about factors that modify dementia risk for adults with Down syndrome. So what is Alzheimer's disease? It is the most prevalent of many causes of old age-associated dementia. It produces devastating neuropathology throughout the brain, resulting in catastrophic cognitive and functional losses. On the left of this slide is a normal elderly brain obtained at autopsy. On the right is a brain from a person with advanced Alzheimer's disease, and it's quite clear that there has been tremendous atrophy associated with this disease. There are many other causes of old age-associated dementia. As you can see from this pie chart, Alzheimer's disease is the most prevalent of the old age-associated dementias, accounting for about 50% of cases by itself, and another 15% where other types of neuropathology are present. Pathological characteristics include the deposition of amyloid beta and neuritic plaques, which Dr. Lott just told you about, neurofibrillary tangles, which Dr. Lott just told you about. There's changes in blood flow and glucose utilization within the brain, and again, eventually leading to loss of brain cells and brain mass, and a whole cascade of neuropathology that we don't need to talk about today. In terms of biomarkers, this is a model of progression of AD as detectable by currently available methods that Jack published in 2013. And if I can work the cursor, which I can't, there we go. You can see some accumulation in CSF of A beta. The red line would be amyloid in the brain as detected with PET imaging, and then you can see CSF accumulation of hyperphosphorylated tau, and then changes in volume and brain structure and glucose utilization, and then eventually cognitive impairment. So all of these now detectable markers of disease give you opportunities to identify prodromal disease and possibly intervention to prevent the dementia that will occur relatively late compared to the changes in amyloid processing and in tau processing that leads to neurofibrillary pathology. This is sort of a rough diagram of clinical progression of an individual that would have Alzheimer's disease. With or without Down syndrome, the progression looks pretty much the same. For a good long time, many years through adult life, you're showing subtle changes in cognition. I'm looking for how many gray-haired people we have in the audience. For those of you who were there, you will know that there were things you could do when you were 25 that you can't quite do as fast now. That is normal. But at some point in time, this, subtle declines with normal aging start to accelerate, and you will hit a criterion that clinically is now defined as mild cognitive impairment, MCI. This is not dementia. It is sort of a pre-dementia, but it's not asymptomatic, where you'll get, changes in cognitive tasks that place a high load on your mental processes. You can see some changes in what are called instrumental activities of daily living. Hardly ever will you see changes in activities of daily living, like self-care. But, you will notice that things are not the same. You'll probably go to your doctor. You'll say, gee, doc, what's up? The doctor will give you, if they're good, some kind of psychoneurological functioning assessment. You'll score fine, maybe a little bit of impairment, but nothing, clinically significant. Then the disease continues to progress, and at some point in time, you will hit a criterion that essentially says you have dementia. Changes in functional ability, inability to perform at work, inability to balance, family finances, and you have dementia. And then eventually the disease continues to where there's devastating impairment, no longer walking, no longer talking, no longer recognizing who anybody is or where you are. At this point in time, of course, diagnosis, you don't need to be a rocket scientist to know that the person is quite impaired. And again, eventually it leads to death. So we've known for a very long time that Down syndrome and Alzheimer's disease, there seems to be some association. In fact, before Alzheimer discovered Alzheimer's disease, there were observations of a change in elderly people with Down syndrome that looked like this premature aging and senility. It's noteworthy that those people back in 1876 were in their 30s. And as you will see shortly, dementia today in the 30s is quite rare for people with Down syndrome. And then, eventually in 1925, the increased risk for AD neuropathology was identified in older adults with Down syndrome, and then Jervis essentially associated that neuropathology with frank dementia. So why is there this association? The dominant kind of mechanism, etiological mechanism for Alzheimer's disease today is the amyloid cascade hypothesis, where the accumulation of beta amyloid in the brain kicks off this whole cascade of neuropathology, and that, of course, is the primary driver of the decline in cognition and function. Well, it turns out beta amyloids form from a large transmembrane protein, amyloid precursor protein. Dr. Lott mentioned this, and this is a protein needed for normal cell functioning throughout our lives. It turns out that that gene is located on chromosome 21, and right there. As you know, people with Down syndrome have 3 copies of chromosome 21, thus 3 copies of the APP gene, except in the rare case that Dr. Lott just described, and V. Prosher also described a very rare case with partial disomy focused on the APP gene. This results in overexpression. Overexpression contributes to increased risk for Alzheimer's disease because it's the metabolism of the APP gene that creates the amyloid beta that coalesces into the plaques that Dr. Lott described. So, general facts about Alzheimer's disease in adults with Down syndrome. Again, as you have just heard, almost all adults over 35 have the neuropathological characteristics of Alzheimer's disease. Incidence of dementia, as you will see shortly, increases in the late 40s and throughout the 50s, as opposed to the general population, where you get an increase in dementia risk in the beginning in the late 60s and then continuing throughout the late 80s. There are many similarities to adults without intellectual disability with respect to affected areas of cognition and functioning and progression of decline. Progression's a little bit faster in adults with Down syndrome, as we have seen it There are some reports in the literature of a very aggressive progression of disease. We think that's the exception rather than the rule. Certainly, if you develop seizures, you see an accelerated progression. But as Dr. Lott just told you, that's sort of this perfect storm of, like, you know, two types of neuropathology going on. There are many of the same risk factors, and many individuals with Down syndrome do not exhibit indications of dementia, and there is substantial variation in age at onset. So this is our most recent data on cumulative incidence. This is the proportion of individuals that will be affected by a particular chronological age. What you can see is before 50, and we have tracked people from like age 30 onward, dementia affects about 5% of the population of adults with Down syndrome. Then you get this pretty linear increase up through the late 60s, but even at age 80, and again, this is very small numbers of individuals, when you get to these older ages, you're still not hitting 100% affected cases. Should say that 99% of our sample is full trisomy 21. So everybody is characterized cytogenetically. Why is this an important issue now when it wasn't, let's say, 20 years ago? That's because there's been dramatic increase in life expectancy for people with Down syndrome, and that makes Alzheimer's disease a serious public health concern. These are data published by the Center for Disease Control back in 2001, and it's not quite life expectancy. This is median age at death. And as you can see, circa 1970, median age at death for individuals with Down syndrome was under 5. By the time we hit the late 90s, it's now up to 50. In fact, projected survival is increasing by 1.7 years per year for adults with Down syndrome. 2 points. The first is this is a tremendous achievement. The second is that if we're looking at what goes on with aging in adults with Down syndrome today, a 60-year-old person with Down syndrome was born circa 1950s. 1950s, when that person was born, their life expectancy was under 10. So the elderly population with Down syndrome today could be very different looking than the elderly population with Down syndrome in 20, 30 years from now. And this is something that we need to continue to monitor. So studies of life expectancy, you can see that in 1955, under 5, by 2004, we're up to 60, and again, increasing continuously to this day by, 1 point something years per year. Public health importance. If you sort of take current demographics, so it's the current world population, or at least the current population in the US and the EU, current birth incidence of Down syndrome, we expect about 500,000 plus individuals with Down syndrome over the age of 55 when this current cohort is that old. That's 140,000 people likely to develop dementia. And if you think of, current costs in the US for serving this population and extrapolate that to the EU, and I know the costs are very different in the US and the EU, but, you're looking at a lot of money. So because of this, there have been a lot of major initiatives focused on lifespan development and AD in adults with Down syndrome. Is somebody going to cue me with time? Because I tend to talk. The Alzheimer's Association has established an international society to advance Alzheimer's disease research and Down syndrome specifically. The International Association for the Scientific Study of Intellectual Disability has a special interest group on aging. The NICHD is establishing a Down Syndrome Connect online registry with components that are focused explicitly on issues of aging. There is a center at the University of Illinois at Chicago, specifically focused on adult aging with Down syndrome, that has a lot of information available for caregivers and clinicians. The American Academy on Developmental Medicine and Dentistry has a new initiative, and they've recently put online a new instrument to screen for dementia in individuals with Down syndrome, probably with other forms of intellectual disability. And then the Wellcome Trust Initiative just had a big meeting in Cambridge, and they're looking to establish international collaborations focusing on understanding Alzheimer's disease and Down syndrome from molecules to cognition. And then there's our group. So some of the many approaches have been summarized in a special issue of Developmental Disabilities Research Reviews, which is specifically adult aging, and Marcia Seltzer and I edited this volume. There's one review specifically focused on Down syndrome. There's another review specifically focused on diagnosing dementia in individuals with intellectual disability. And then V. Prosher has published a recent book, Neuropsychological Assessments of Dementia, Down Syndrome, and Intellectual Disabilities. And there are reviews of specific instruments that people are playing with. So, to our program. This is a collaboration between the New York State Institute for Basic Research in Developmental Disabilities, which is a component of New York State government located on Staten Island, which is the southernmost borough of New York, also the location of Willowbrook way back when. And then we have people who join us at Columbia University, and then myself and a few colleagues at the Kennedy Krieger Institute and Johns Hopkins University. This is our group. I won't go through what everybody does. I just want to make sure that you all know that it's a lot of people who are contributing to these things that I'm going to be discussing today. We also have over 100 program sites cooperating with us. We've had to date over 600 individuals with Down syndrome that have volunteered to be in our studies, and we've had generous support from the National Institutes of Health as well as New York State government. Overall aims, we study developmental aging of adults with Down syndrome. Before, we also studied aging and dementia in other populations with intellectual disability, but we're not doing that anymore, mostly because in our hands, their aging looked very much like everybody else's aging. And of course, we study Alzheimer's disease, otherwise I wouldn't be here talking to you today. And we study lots of aspects, the genetics of individual differences, progression of disease, cognitive assessment, neuropsychiatric changes, biomarkers, but mostly I'm going to talk today about diagnosis and individual differences. So, cumulative sample, 600 plus individuals with Down syndrome, IQs from very low to very high. Ages from under 40 to over 80, and that's Down syndrome adults. We test people every 14 to 22 months in this big battery, and it includes a comprehensive chart review, every test, every diagnosis, every medication that they're taking, and every caregiver concern. We interview caregivers that are knowledgeable about these individuals. That's a whole bunch of different things. It takes us about an hour and a half working with the caregiver. We have face-to-face individual cognitive assessments. It's about 2 hours of testing broken up into usually 2, sometimes 3 sessions, depending upon whether the individual is hanging with us or not. I should say, in addition to the data from the cognitive test, we have ratings of whether the person was engaged in the assessments. So we also videotape all of the assessments, so we know that if somebody's starting to show declines, we can see if it's because they were just not interested anymore, or if there was something funny about the testing situation. The assessment includes 3 different measures of mental status, a modified version of the mini mental state exam, the test for severe impairment that Marilyn Albert and her colleagues developed that was looking for assessment of progression in advanced dementia from the general population, and then another assessment that just gives us converging information from the other 2. We look at verbal fluency with the standard method, visual motor integration task, which is a standardized assessment that involves paper and pencil copying of figures that increase in complexity, block design from the WISC-R, supplemented with some easier items, and then a bunch of memory tests. And then we do blood sampling for biomarkers and for karyotyping and for looking at genetic factors that could modify risk. So every cycle of this assessment, we have a consensus conference where we consider every bit of information we have about everybody, and we classify their dementia status as either not demented, with mild cognitive impairment, which is sort of, we can't say that they're not demented anymore, but they're certainly not demented. So it's like this transition status, and that's very much what mild cognitive impairment in the general population is also. And then, if there are clear declines from what we saw before, then they're classified with possible or definite dementia. The basic difference is the first time we classify them with dementia, it's almost always possible, and then if we see further decline, it shifts to definite. I should say that we've never had a person who we classified as possible dementia get better. And then there are a couple of people who were so low functioning when they entered the study that we just, were a little shy about saying, yes, they're declining. That's this unknown status group, and it is literally a handful of people. And then with complications, things happen. These are biologically elderly people. They fall. They have a change in their life circumstances, which we heard from this morning can have an impact on their, cognitive performance. So if any of that goes on, we sort of set that aside and we come back the next cycle to see, sort of if their decline in performance had resolved or continued to get worse, where that, sort of acute incident was resolved. And then they'll get classified again as either one of the 4 categories in yellow. So how can we recognize MCI or dementia in adults with Down syndrome given their pre-existing cognitive impairments? Because we know the performance will be well below population-based criteria on standardized assessments prior to their decline. So all of those assessment batteries that are, sort of appropriate for adults from the general population really just don't work. A lot of times, the items are too complicated to expect an adult with Down syndrome to do them in the first place, like, count backwards from 100 by 7s. And then, of course, performance is expected to vary substantially from person to person. As I said, we have some people with, quite profound intellectual disability and some people with quite mild intellectual disability. And then performance in cognitive tasks is notoriously variable over time for individuals with intellectual disability, and you have to be able to, sort of understand that. So the bottom line is, if this is your normal population, not demented, and this is your demented population, if you have an instrument that can tell the difference between those 2 conditions, big deal, because they are just profoundly different. What we want is to develop instruments that can tell the difference between normal aging and this early changes. You can call it mild cognitive impairment, you can call it early dementia, because as treatments become available to slow the progression or halt the progression of Alzheimer's disease, they're going to be effective early, because over here, the brain damage has already happened and it's not going to be reversible. So what's the best practice? There is no current consensus on methods or criteria for diagnosing dementia, or MCI for that matter, when it occurs in adults with intellectual disability. The accepted best practice, we think, is to document clinically significant declines for the individual patient, but evidence-based validation of specific definitions of clinically significant decline have yet to be done for adults with Down syndrome. So we need to determine which changes are truly reflective of AD progression and which are not. And we need to evaluate both sensitivity and specificity. A lot of people are very focused on sensitivity, that is, identifying a case with dementia when it's really there, but not so much focused on specificity, which is not over-identifying dementia in people when it does not, in fact, exist. So we looked at this, and we assessed this large number of elderly adults prospectively. We looked at changes and quantified them over one assessment cycle, which in our case is 14 to 22 months. And then we wanted to relate specific changes to gold standard dementia status, which in our case is this consensus determination based upon everything we know about these individuals, and then determine if observed declines tend to be followed by further declines. So if we say you are demented and we set up a criterion for a specific instrument in terms of a decline, if you truly have dementia, when we come back and look at you in another 14 to 22 months, you should show further decline. On the other hand, we don't know that that's what happened, so this is what we looked at. So we started out looking at instrument by instrument, and I'm going to talk about the DMR, which is now the DLD, that's Dementia Questionnaire for Individuals with Learning Disability, because it's published in the UK, and in the UK, intellectual disability is called learning disability. And again, Helene Evenhuis and her colleagues did this. This is sort of an industry standard informant interview now, and it generates two scores, some of cognitive scores and some of social scores. And according to the manual, if you show okay, so this is, the higher your score, the worse you are in terms of status, but I've transformed these data to show the lower you are, the worse you are graphically. But the bottom line is if you show a change in 7 or more points on the sum of cognitive scores, she has shown that it has very high sensitivity and acceptable specificity for dementia. If you show a change of 5 points on the sum of social scores, same thing. It's predictive and diagnostic of dementia as a screen. When we looked at this, we found that sensitivity for her published criteria are not very good. Specificity, however, is very comparable to what she showed in her manual. And the reason for this is these summarized here. And I'm going to walk you through this graph. So this is showing a 7-point drop in the DMR, which is again a 7-point increase, but we're calling it a drop because it shows that you're worse clinically. And this is showing change from time 1 to time 2. And over here, we're plotting time 2 to time 3. So the gist of it is, if you are truly declining, if you truly have Alzheimer's disease and this instrument is working the way it's supposed to, once you show a change of this magnitude, when we come back and look at you again, you ought to be all in this quadrant. That is, you don't get better. To the degree that you fall in this quadrant, it says you got better. If you have Alzheimer's disease, you never get better. So, the bottom line is, if you meet this standard as published, you're just as likely to get better as get worse, and that makes it a not very good instrument in and of itself. Now, there's some signal in here But the bottom line is the simple measure of change is not particularly informative as a single assessment. And again, there's nothing special about this assessment. This pretty much fits the bill for just about all of the assessments within our battery. These are the same data for the sum of social scores, and you can see again exactly the same profile. All of these points ought to be down there, and you can see that that's simply not the case. So, quantification of decline, they've got to be interpreted with considerable caution. Some measures seem to be more informative than others, but the bottom line is no one assessment by itself has accuracy high enough to inform clinical diagnosis at this point. Overall findings highlight the complexities of interpreting declines within an individual patient. I should say the group data look fantastic. So if you have change over time for the group of individuals with dementia versus the group of individuals with MCI versus the group of individuals with no dementia, you see exactly what you think you ought to see. More decline for the group with dementia, intermediate decline for the group with MCI, no decline for the group of individuals without dementia. So, are we stuck? And the answer is no, we're not stuck. We can always figure out something. So, we came up with an alternative strategy, and that's to assess a large number of elderly adults prospectively, do the same classification of their dementia status, but determine one-time measures of performance anchored to premorbid IQ. And we chose premorbid IQ because we had that information on our individuals. It could be anchored to premorbid, Vineland score, premorbid global cognitive ability score. You pick, sort of what works for you. And when we do this, we come up with data that looks something like this. So all of those dark dots are individuals with dementia. The red triangles and the black circles, and all of the blue circles unfilled are individuals without dementia, and all of the red squares unfilled are individuals with MCI. And what we've plotted here, again, are the DMR, DLD, sum of cognitive scores against Stanford Binet IQ measured early on in adulthood or in childhood. And what you can see is that, again, lower scores are good here. The higher IQ, the lower your score, which is the more cognitive ability you have. And what you can see is that generally, people with dementia score way higher, i.e. way worse than people without dementia. And in fact, we've imposed a criterion area here where if you are above the top green line, you have dementia, and if you are below the bottom green line, you do not have dementia. And in between the lines, we're saying we don't have the precision to, know what's going on exactly. And those two lines are separated by one standard error of measurement. I lost the sensitivity and specificity. So, just eyeballing it, I don't remember what the exact numbers are, but both sensitivity and specificity are around 0.8 or higher. You can see that there are a couple of red triangles in the not demented range, and there are certainly some blue circles in the demented range. But the bottom line is, you're going to not be, perfect. If you shoot for perfection, you're going to be very disappointed. Another scale is the basically a modified version of the mini mental state exam that was, sort of the industry standard for a direct face-to-face screen. It takes about 10 minutes to do or less. You can do it at the bedside of your patient. You're looking at orientation to person, place, and time. You're looking at, some measure of executive process, either, again, count backwards by sevens from 100 or spell the word world backwards. Repeat sentences, the most complex one being repeat after me, no ifs, ands, or buts. I wish I could say that in French, but I apologize. And then there's copying a fairly complex design. And the bottom line is if you're a healthy, normal adult, you'll score virtually perfect on this test. And if you are demented, you will score below 24 out of a possible 30. So we modified this, and I'm just putting this up with the same IQ-anchored scoring method to show you that this works the same as it did for the DLD. Again, the better you are, the higher you score, and you can see that pretty much almost all of the blue dots are above the green lines, and almost all of the solid symbols are below the green line. Sensitivity, specificity, you can see that if you think of -2 standard errors of the mean below that criterion score, virtually none of the not demented people score that low. If you think of above 2 SEMs, virtually none of the demented people score that well. So this is working pretty well. Biomarkers, do I have time? Okay, so I will just say that, lots of people are looking at biomarkers because if you can come up with a good biomarker of dementia status, you avoid all of the complications that I just reviewed about trying to come up with a clinical quantitative assessment of change in cognition. Blood biomarkers have tremendous advantages because you can just, stick a needle in somebody's arm and get some blood. Much easier than a lumbar puncture, much easier than imaging. The disadvantage is that blood is not brain, and it's sort of, what's going on systemically does not provide direct indications of what's going on within the central nervous system. There are a bunch of blood-based biomarkers under study, and for the general population, as Dr. Lott just mentioned, there's a new RFA, a request for applications that has been issued by the National Institute of Health to come up with biomarkers for adults with Down syndrome. We've looked at A-beta in blood. Again, the mean values look fantastic. The variability of across individual patients make it really difficult to use it clinically as a biomarker. But we've also looked at telomere length, and Ed Jenkins is our lead investigator with this. Telomeres are the ends of chromosomes. Each time you get cell replication, the telomeres get a little shorter in people and other mammals and other eukaryotic organisms, and eventually it leads to cell senescence, and this would be, sort of the cell biology mechanism of aging. So this is, you can tag these telomeres with a fluorescent tag, and you can actually measure the length of these things mechanically. You can measure how bright the tags are as a way to quantitate it, and what we found was that, in fact, this works out as a very robust and impressive biomarker of dementia status in adults with Down syndrome based on a small study of 11 patients with MCI and 11 patients controls. So we're very interested in pursuing this. This essentially shows the telomere length of controls versus telomere lengths of patients. And again, this is just different ways to measure it. Makes a little bit of a difference exactly what your procedure is, but the bottom line is, big differences. We wonder if these data are a little too good to be true, but that didn't stop us from publishing them. And hopefully, when we put in a grant to replicate these findings, our funding agency will think it's worth pursuing. Factors modifying risk, I'm going to go through this very quickly because I seem to be running long, and I was told that that was going to happen, and I ignored that because I thought I would talk fast. So estrogen, bioavailability of estrogen tends to be good. This is cumulative incidence of dementia in women with Down syndrome with high bioavailability of estrogen versus lower bioavailability of estrogen. And you can see that dementia seems to be occurring in the women with low bioavailability of estrogen roughly 5 years earlier than in women with high bioavailability of estrogen. So, I mean, you know, suggests that maybe hormone replacement trials for women with Down syndrome perimenopausally might be therapeutic. We also see, we've looked at cholesterol, and what we find is that people with high cholesterol, total cholesterol above 200, tend to have onset of dementia earlier than people with good cholesterol. And that is corrected when you take statins. So if you have individuals with Down syndrome with high cholesterol, it might be a good idea to treat them. Other factors, I mean, this is general population data that have been played with. Physical activity seems to be the most protective because it increases brain oxygenation. But there are other things that are being tried all the time. But physical activity seems to be the best in terms of the strongest and most consistent evidence of benefit. So what should clinicians do? Be vigilant regarding changes in memory orientation and new indications of psychiatric concerns. Repeat assessments of functional skills and cognition that you've done previously, and understand that risks for all aging-related conditions are there, and consider the possible impacts of changes in health, sensory status, as Dr. Lott told you, or the social environment, as you heard about this morning. And I think I will end with just a hope for the future. This is a figure I stole from Steve Dukowski, and each one of these dots is 100,000 cases of Alzheimer's disease projected by the year 2050. So the bottom line is we can piggyback on the interest in Alzheimer's disease that's out there worldwide in terms of attracting additional research resources to study aging and dementia in adults with Down syndrome. And thank you very much.